Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the Histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below.
Hi! In this first of the two endocrine lecture series, we'll discuss the anatomic overview of the endocrine cells and organs and spend some time learning the developmental, physiological, and histological organization and function of the pituitary gland. Endocrine system as a whole is comprised of a number of organs and many individual cells and cell clusters that release hormones into the circulatory system, basically into the bloodstream, so that the hormones may travel throughout the body and affect the distant target cells or organs. The major organs of the endocrine system, going from top to bottom from anatomical perspective, includes the pituitary gland. The pituitary gland is interesting in that it is comprised of two different types of tissues. One is an extension of the brain itself, and the other a proper epithelial-derived endocrine structure. The pituitary gland is also called the hypophysis because anatomically speaking, it is positioned on the underside of the brain structure. The pineal gland is actually an extension of the brain tissue in the posterior direction. I know this diagram could be a little confusing in that we're looking at the anatomy from the anterior aspect, but this diagram of the brain is actually on the sagittal plane. At any rate, this pineal gland, which is also called the epiphyses, is responsible for producing melatonin, which controls our circadian rhythm. Next, we have thyroid. Thyroid is a butterfly-shaped organ that sits just under the larynx and in front of the trachea, and it produces important thyroid hormones, which influence our metabolism, our growth, mental state, many different functions. Behind the thyroid, on the posterior aspect, there are parathyroid glands. So from this anterior aspect, parathyroids are not diagrammed, but if I were to point them out, they would be positioned about there. Parathyroid glands produce the parathyroid hormones, which influences our blood calcium level. You may recall from the bone tissue lecture. Then we have the adrenal glands, which are these pyramidal-shaped organs positioned just above the kidneys. These paired adrenal glands produce many hormones, including those adrenalins. The ovaries and testes are also endocrine organs, which produce pretty important sex hormones. Lastly, there's this concept of diffuse endocrine system. This system refers to each individual unicellular endocrine cells and cell clusters that are distributed throughout our body that release hormones. So a good example of the structure of diffuse endocrine system would be the pancreatic islets or islets of Langerhans that was discussed in the digestive system lecture, which secrete insulin, glucagon, and somatostatin. Another good example of diffuse endocrine system includes the enteroendocrine cells, many of which are unicellular endocrine cells that are distributed throughout the GI tract, releasing their products into the bloodstream. There are other unicellular endocrine cells distributed throughout other organ systems. In this particular lecture, we'll look at these major organs of the endocrine system, since we've already discussed some great examples of diffuse endocrine system, and the ovaries and testes will be discussed in the respective reproductive system lectures. Anatomically, the pituitary gland, also called the hypophysis, is a really small and ovoid and fleshy structure that sits within this bony casing called the sella turcica. In this diagram, the area containing the pituitary gland has been blown up right here. We can see this pea-sized structure sitting within and encased by this bony structure. This bone is the sphenoid bone, and this depression within this bone is called the sella turcica. It literally translates to a Turkish saddle. So we can see that the pituitary is a pretty stationary structure that is well covered and protected by the bony structure. Physically speaking, the pituitary gland is connected to the hypothalamus by a thin stalk called the infundibulum. And what the infundibulum attaches to is a structure of the brain called the hypothalamus. So based on this close anatomic relation between the hypothalamus and the pituitary gland, these two structures function very, very closely together as well. We'll see that later in this lecture. The pituitary gland is also really interesting in that it develops from two embryonic derivatives, which results in two different types of tissues that comprise this ovoid organ. One type of tissue is the anterior pituitary. This is a pituitary gland with anterior side and posterior side here. The anterior aspect of this pituitary is also called the adenohypophysis, with the adeno referring to the gland. So this is the part of the pituitary that is consistent histologically with any endocrine glands, in that they derive from the epithelial cell ingrowth into the underlying connective tissue, which then loses its connection or the duct to the originating covering epithelium. So the anterior pituitary develops this way as well, and where it develops from is the oral ectoderm. So if I were to draw an oral ectoderm in there, or the oral cavity, there would be the lining epithelium here. So imagine some of these lining epithelial cells thickening and growing towards the developing brain and eventually becoming pouch-like, at which point this ingrowth is called the Rathke's pouch. And this ingrowth will eventually go up there and fuse with the neural tissue that's coming from the hypothalamus and lose its connection to the oral ectoderm. So voila, now we have an endodermal organ that is truly derived from an epithelial tissue. That's how the anterior pituitary forms. The posterior pituitary, on the other hand, is also called the neurohypophysis for a reason. This is the part of the pituitary gland that is essentially an extension of the neural tissue from the hypothalamus. So if this is the hypothalamus, we have neuron cells and their axons that are all growing down, and their axon terminals that are all occupying this region, thereby giving rise to the posterior pituitary. Clearing all of those doodles and showing you the actual histology of the anterior pituitary, from this macroscopic view, we can easily see the tissue staining differences between the adenohypophysis and neurohypophysis. This two staining pattern is based on the tissue makeup of the anterior and posterior pituitary glands. The posterior pituitary gland being essentially an extension of the diencephalon, a region of the brain. It is comprised of the neural tissue, and as we zoom into the posterior hypophysis, we'll see more and more resemblance to the neural tissue histology. Whereas the anterior pituitary, we will likely see the histology that is more consistent with epithelial-derived glandular cells. And as mentioned earlier, the pituitary gland is structurally and functionally connected to the hypothalamus. Now, considering that the pituitary gland is an endocrine organ which releases its hormones into the bloodstream, it is very important to understand that there are two different vascular supply that support the tissues and functions of the pituitary gland. One is the superior hypophyseal artery, which supplies the hypothalamic hypophyseal portal system. To explain the hypothalamic hypophyseal portal system in a little more detail, the superior hypophyseal artery will come in and branch into the rich fenestrated capillary networks here in a region called the median eminence, which is a distalmost region off of the hypothalamus. Within the hypothalamus, there are a lot of these neurons that are synthesizing the regulatory hormones for the cells in the anterior pituitary. And all of these regulatory hormones are then transported through these neurons and are released at their axon terminals, which are positioned within the median eminence. So as these regulatory hormones are being released into the first capillary network, these regulatory hormones are then carried away from the median eminence into the anterior pituitary by a hypothalamic hypophyseal portal vein. And within the anterior hypophysis, this portal vein once again branches out into the fenestrated capillary networks all throughout the anterior pituitary. What happens here at the secondary capillary networks is all these regulatory hormones that's been picked up from the median eminence are stimulating or inhibiting the cells of the anterior pituitary gland. And in response, the cells within the anterior pituitary are now releasing or stop releasing their hormones. And a lot of these released anterior pituitary hormones are then being carried away by the hypophyseal vein to the systemic circulation. The second vascular supply is the inferior hypophyseal artery. It primarily comes in and branches out into fenestrated capillaries within the posterior hypophysis or the neurohypophysis, picking up any hormones that are released from the posterior pituitary and carrying them out and draining into the hypophyseal vein. And although there are some anastomosis between the superior and inferior hypophyseal arteries, for the most part, it's quite remarkable to think that the anterior and posterior pituitary have relatively independent vascular supply. To summarize the hypothalamic hypophyseal portal system, it refers to the two capillary beds that are connected by a portal vein. This entire system is primarily supplied by the superior hypophyseal artery, which comes in, branches into the first capillary bed or network of fenestrated capillaries in the median eminence of the hypothalamus. And this is where the regulatory hormones from the hypothalamic neurons are released and picked up by these capillary networks. All of these regulatory hormones are then carried away by the hypothalamic hypophyseal portal vein, which then drains into the second capillary bed positioned within the anterior pituitary. Here, the regulatory hormones that were picked up by the first capillary bed bathe the endocrine cells of the anterior pituitary cells, which in return release their own hormonal products into the second capillary bed, which is picked up by the very same second capillary bed network, which eventually drains out of the pituitary by the hypophyseal vein and into the circulatory system. So this is quite the sophisticated system of regulation using circulatory system between the hypothalamus and the anterior pituitary. The posterior pituitary too has an intimate connection with the hypothalamus. We have to remember that the posterior pituitary is an extension of the neural tissue from the hypothalamus. Specifically, there are two regions within the hypothalamus that host the cell bodies of the neurons that extend their axons down into the posterior pituitary. So if we were to look at this diagram at a higher magnification, and let's say that this is the hypothalamus, this is the infundibulum, and posterior pituitary down here, then anterior pituitary would be its own kind of entity right here. One of these two nuclei in the hypothalamus is called the paraventricular nucleus. And here, the cell bodies of the neurons are synthesizing these hormones called oxytocin, which has been associated with feelings of love and pair bonding. And the other nucleus in the hypothalamus is physically positioned just above the optic chiasm. So this nucleus is called the supraoptic nucleus. This structure too hosts a lot of the neuron cell bodies that primarily produce the hormone called the vasopressin, which is also known as the antidiuretic hormone or ADH. Once these hormones are synthesized within the neuron cell bodies, they are then transported through their axons, which travel through the infundibulum down into the posterior pituitary, where the axon terminals branch out within the posterior pituitary. So similar thing would be happening here with the neuron cell bodies that are positioned within the supraoptic nuclei. This collection of axons coming from the paraventricular nuclei and the supraoptic nuclei collectively form this infundibulum, also called the hypothalamic hypophyseal tract, with the tract being a generic term referring to the collections of axons that travel together within the central nervous system, just as the nucleus or the nuclei is a generic term referring to the the collection of neuron cell bodies within the central nervous system. And cytologically, the hormones that are synthesized within the neuron cell body are being transported along the length of the axon by the kinesin along the microtubule cytoskeletal network. The axon terminals in the posterior pituitary then store the hormones that have been transported down here, and sometimes these axon terminals can get dilated quite a bit so that they're readily visible under the microscope, in which case these dilated axon terminals are also called the herring bodies. So the hormones that are released from the posterior pituitary are ADH or vasopressin, which is synthesized in the hypothalamus by the neuron cell bodies that are positioned within the supraoptic nuclei. The ADH or vasopressin have their receptors in the kidneys, in the collecting tubules and ducts of the kidneys. In response to ADH, the collecting tubules and the ducts within the kidneys will increase the water resorption, thereby retaining water within the body. The other hormone secreted by the posterior pituitary gland is the oxytocin, which is involved in generating the feeling of love, pair bonding, as well as uterine and mammary gland contraction, which aids in childbirth and lactation. From the histological perspective, not only can we divide the pituitary gland into these big anterior pituitary and posterior pituitary or neurohypophysis, but also we can subdivide each of these big lobes into smaller and specialized regions. The anterior pituitary can be divided into 3 subregions. The pars distalis is the biggest portion of the anterior pituitary. The pars intermedia is the portion of the anterior pituitary that abuts the posterior pituitary immediately, like so. And the pars tuberalis is a small portion of the anterior pituitary that kind of wraps itself around the infundibulum. The posterior pituitary is subdivided into 2 regions. The pars nervosa refers to the entirety of the posterior pituitary lobe, and the pars infundibulum refers to the infundibulum, this neural stalk, also known as the hypothalamic hypophyseal tract. Focusing our attention on the anterior pituitary histology, at a higher magnification, the glandular nature of this organ becomes a little more apparent in that it is quite cell-rich with this variety of cells that are forming these aggregates of cords or plates or nests of cells that are surrounded by rich network of fenestrated capillaries. This anterior pituitary is comprised of 2 types of endocrine cells, the chromophils and chromophobes. Chromo refers to color. Phils refers to philia or love. So these are the cells that love color. They stain either eosinophilic or basophilic. Phobes or phobia refers to fear. So these are the cells that fear the color. So these are the cells that do not stain and instead appear pale in regular H&E staining. Of the chromophils, there are 2 subtypes of cells. One is the acidophils, and as the name suggests, these are acid-loving cells or acidic dye-loving cells. So the acidophils stain pink to red in their cytoplasm, and we can see in this field of cells that there are numerous pink or eosinophilic staining cells all strewn about. The acidophils throughout the anterior pituitary produce 2 specific hormones, the growth hormone or the GH and prolactin. The growth hormone, of course, is responsible for cellular growth and regulating or increasing our metabolism, whereas prolactin is involved in mammary glandular development and milk production. The basophils, on the other hand, are the cells that love basic dye, such as the hematoxylin. So these are the cells that are staining more bluish to purple in color throughout this field of cells. These cells collectively produce 5 different anterior pituitary hormones. The MSH, or melanocyte stimulating hormone, as the name suggests, stimulate melanocytes throughout our body to produce more melanin or skin pigment so we can get darker as a result of MSH increase. The FSH is a follicle-stimulating hormone, which stimulates the production of ovarian follicles. The LH, or luteinizing hormone, also has its target cells in the ovaries in that it triggers the ovulation of the largest of the follicles in the ovaries. The ACTH stands for the adrenocorticotropic hormone. As the name suggests, ACTH stimulates the cells of the adrenal cortex, especially a region of the adrenal cortex called the zona fasciculata, which in response to ACTH releases more cortisol, which is a stress hormone. We'll revisit ACTH as we discuss the adrenal histology. And lastly, we have TSH, or thyroid stimulating hormone. And again, as the name suggests, this hormone would stimulate the cells of the thyroid and increase their thyroid hormone secretions. The chromophobes, again, are the cells that fear or that don't stain well with either eosin or hematoxylin. So these are these pale staining cells, at least in their cytoplasm, that are strewn about. And these chromophobes are actually considered to be either acidophils or basophils that have already released their products into the bloodstream. Therefore, they're appearing pale. And some also suggest that the chromophobes are the undifferentiated stem cells of the anterior pituitary that can eventually differentiate into either acidophils or basophils depending on the functional needs. The histology of the posterior pituitary is quite different than the anterior pituitary, which again we can appreciate just from the macroscopic observation as well. It stains quite pale and, relatively speaking, doesn't seem as cellular. And this is because the posterior pituitary is really an extension of the hypothalamic neural tissue. And as mentioned earlier, the posterior pituitary is comprised of two subregions, the pars nervosa, which hosts a lot of these dilated axon terminals of the neurons whose cell bodies are positioned within the paraventricular and supraoptic nuclei in the hypothalamus. These dilated axon terminals containing oxytocin or vasopressin are called the herring bodies. At a higher magnification of the boxed area, we can appreciate some of these herring bodies by these amorphous, somewhat eosinophilic, staining, dilated structures that are occasionally observed. Also found within the pars nervosa histologically are the astrocyte-like glial cells called the pituicytes. So a lot of these elongated to oblong nuclei belong to the pituicytes, which function very similar to the astrocytes within the central nervous system. Lastly, we see a lot of axons that are traveling throughout. So these axons, as well as the glial cell processes, are forming the all too familiar neuropill of the pars nervosa. The pars infundibulum is essentially the infundibulum, which contains collections of the axons. At a higher magnification of this box up here, we'll see the histology that is quite reminiscent of the tracts within the central nervous system, with axons that are traveling in one direction together. And since these axons are traveling between the hypothalamus to the hypophysis or the pituitary gland, calling this the hypothalamic hypophyseal tract would be entirely appropriate. The axons that travel within the pars infundibulum are unmyelinated axons, so little to no oligodendrocytes here.
Thank you for watching this video from the Da Vinci Academy Histology video course, which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.